Imagine an organism capable of taking foreign genetic material, flash freezing it, and keeping it locked inside a biological vault for years until the exact moment it is needed. Finding a mate in the undergrowth is a dangerous gamble that exposes both individuals to predators. For many species, traditional mating is a high-risk liability that requires a specialized anatomical workaround. This document outlines the core functions of the insect reproductive system, and the first item on the list is the continuation of the species. Every other biological process serves this single requirement. Most insects follow a dioecious model, meaning the male and female reproductive systems are housed in entirely separate individuals. Because finding a partner in a hostile world is rare, insects have engineered internal hacks to guarantee successful reproduction from a single fleeting encounter. Operating as separate entities subjects males and females to different survival pressures, leading to sexual dimorphism, where the two sexes evolve drastically different physical forms. These two Drosophila fruit flies demonstrate this morphological split. The female on the left is significantly larger than the male on the right. These external physical differences house highly specialized, microscopic reproductive factories operating internally. This diagram of the female reproductive system shows the two massive, clustered ovaries. They're secured to the body wall by thread-like suspensory ligaments at the top. These internal systems occupy a large portion of the body cavity to maximize volume. They are built to mass-produce the next generation rapidly, often before the mother is killed by predators or weather. The ovaries are composed of individual tapering assembly lines called ovarioles. At the top of the tube is the germarium, producing undifferentiated germ cells. As cells move down, they enter the vitellarium to grow into mature eggs. Some insects accelerate this process using nurse cells, or trophocytes. As seen in the rightmost meroistic diagram, these cells pump nutrients directly into the growing egg through cytoplasmic strands. This automated nutrient delivery system allows female insects to produce eggs at a scale and speed that ensures survival despite high mortality rates. To maximize a single mating encounter, the female system includes a specific sac branching off the tract, called the spermatheca. The spermatheca secretes specialized fluids that keep deposited sperm alive and viable for hours, months, or even years. Certain species utilize a double-door system. They feature one opening, the vulva, for receiving sperm, and a separate exit, the ovopore, for discharging eggs. This storage anatomy allows a female to brave the outside world for a mate only once, while remaining capable of producing offspring continuously for the rest of her life. The male's primary challenge is ensuring a microscopic genetic payload survives the transfer into the female's internal environment. Inside the testes, germ cells undergo a transformation. This diagram shows the vertical progression from the top of the tube down to the mature spermatozoa at the bottom. These mature cells pool in the seminal vesicles, acting as a holding tank until they are deployed through the ejaculatory duct. The male system also utilizes accessory glands, seen here at the top of the diagram. These glands secrete fluids that facilitate sperm transmission and provide life support during the jump to the female. The male's anatomy functions as a precise chemical delivery system, providing the specific fluids necessary to keep genetics intact until the transfer is complete. While the standard male-female system is highly efficient, evolutionary pressure has pushed some insects to bypass it entirely. Through a process known as parthenogenesis, female insects have evolved the incredible ability to reproduce entirely asexually, essentially cloning themselves without mating. Aphids use this cloning mechanism to rapidly swarm and dominate plant stems, as seen in this image. Other insects utilize polyembryony, where a single egg splits into multiple embryos, multiplying the output of a single reproductive act. Some species even possess both male and female organs. This functional hermaphroditism allows for self-fertilization, making males unnecessary for the survival of the species. From internal storage vaults to biological self-cloning, these microscopic systems make insects the ultimate biological survival machines. They dominate the Earth because their anatomy is designed for total self-contained resilience.